Na na na. Welcome to the bacteria competition for MYP Biology, fall of 2022. It is great to have you. In today's tournament, we have some serious bacterial competition. But before we get into the matchups, we should go over the rules. When designing their bacterium, students had 3 main tasks. They had to give their bacterium a morphology or shape. They had to decide how it was going to move around in its environment, which is locomotion. And after reproduction by binary fission, students had to decide how their bacteria was going to prefer to exchange its DNA. That would allow for genetic diversity, which would then allow for more successful colony formation. So let's start with morphology. Students had 3 options to choose from. The first was the coccus morphology, which is spherical, round, and fairly small bacteria. Students could also choose to have their bacteria form a bacillus shape, which was more capsule-shaped and rounded. The last option for morphology was a spirochete, which can be thought of as a spiral. For locomotion, students also had 3 options. The first was gliding movement, and with gliding movement, the bacteria in colony formation would be on top of a semi-aqueous or semi-liquidy surface, and they would move as a clump. The second option was flagellar movement, and for a flagellum, it's like a tail structure on bacteria that can whip back and forth in order to propel the bacterium forward. The last option for locomotion was a spiral swim. This one could have been interpreted in 2 different ways. If you have a spirochete doing a spiral swim, it would just be rotating in order to move itself forward. Another option for the students was multiple flagella working in tandem, and these would have coordinated motion, either clockwise or counterclockwise, to push the bacterium forward. The last component of the designer bacterium was how that bacteria was going to adopt genetic variation. We talked about bacterial reproduction through binary fission, and perhaps the best way to think about binary fission is the prokaryotic version of mitosis. The same goal is accomplished where DNA of the bacterium is replicated, and the bacterium divides into 2. These 2 daughter cells are exact replicas or clones of the original cell's DNA, much like the 2 daughter cells from mitosis are going to be exact replicas of the original cell. However, we can't say that bacteria undergo mitosis because they don't have some of the most important structures involved in the process, such as a membrane-bound nucleus, centrosomes that create spindle fibers, and they have a different shape to their chromosomes. So there wouldn't be spindles coming from the centrosomes to attach to the centromeres and pull the chromatids apart. So we cannot call bacterial reproduction mitosis, but you can think about it as having the same goal. After binary fission, however, bacteria do have ways of exchanging DNA in order to promote genetic variation. Genetic variation in bacteria can help them resist antibiotics or could allow them to be more successful in their environment. So the 3 methods of DNA exchange that the students could choose from were conjugation, transformation, and transduction. Conjugation is able to happen through direct links between bacteria called pili. So this tube structure is formed between 2 bacteria, and DNA is passed that way. Another way that bacteria can acquire novel or new DNA is through transformation, where there's some bit of DNA in the environment, and the cell, the bacterial cell, will take it in and incorporate it as part of its own DNA. The last method of DNA exchange was transduction. And with this, you have a bacteriophage, which is a type of virus. It's the one that looks like the lunar lander. I'm going to try to draw one here. And it's when one of these bacteriophages, and this is not at all drawn to scale, transfers genetic material from one bacterium to another bacterium.